Well, I would like to welcome everybody here for today's webinar, and thank Dr. Shanks and Dr. Robinson for their participation here and telling us their story about DUMPS. Dr. Shanks and Robinson, go ahead and tell us the story.
Thanks very much, Eric, and appreciate the opportunity to be part of this team effort for the webinar series. And I'm Roger Shanks, and you have read a little bit about me, but something you might not have known, and that Jim asked me earlier in the week, is whether I had any biochemistry courses. And I have to admit that I had biochemistry as an undergraduate at the University of Illinois, and also I think I had a couple of biochemistry classes from Don Bites at Iowa State as part of my graduate work, in addition to the genetics and all the statistics you need to be having as a quantitative geneticist.
I'm Jim Robinson, and I'm a true biochemist. In fact, I've never taken a course in genetics or statistics. As Roger grew up on a dairy farm, I didn't know anything about dairying until I became a faculty member here at the University of Illinois in the Dairy Science Department. The DUMPS story is a story of true collaboration. Roger and I have clearly complementary strengths and experiences, and facilitated our work in discovering and characterizing and eventually eliminating DUMPS from the Holstein breed. The most satisfying, the DUMPS story is, the DUMPS research is the most satisfying research experience of my professional career, and we both appreciate the opportunity to share the DUMPS story with you today. So let's review what DUMPS is. DUMPS is the acronym for Deficiency of Uridine Monophosphate Synthase. It's an inherited disorder in Holstein cattle, as we showed. It's inherited as an autosomal recessive. In the homozygous recessive state, it leads to embryonic mortality around 40 days into the gestation or thereabouts. Let's recall what serendipity is. Serendipity can be defined as the seeming gift of finding something good accidentally, or alternatively, the making of fortunate discoveries when not in search of them. And that is certainly was the case in the DUMPS story. The term serendipity was coined by Horace Walpole some years ago, 1754, in a fable entitled The Three Princes of Serendip. Serendip was an old name for Sri Lanka, an island off the coast of India. And the heroes, or the princes in the story, go about their journey and make fortuitous discoveries along the way. The serendipitous discovery of DUMPS relates to the fact that we were studying milk orotic acid, and this unexpectedly led us to the discovery of DUMPS. So let's remember what orotic acid is. It's a pyrimidine. It's a carbon-nitrogen ring. It is an intermediate in pyrimidine biosynthesis, and it's present in cow's milk at substantial levels, 0.1% of the solids. Now, that may not seem like much, but recall this is an intermediate of metabolism, and for it to be present at these levels is quite surprising. Let me just mention that most milks contain a dozen or so pyrimidines and purines, in non-ruminant species such as the pig, the horse, and humans, orotic acid is not present at all. In ruminant species, sheep and goats, they have orotic acid in addition to 12 or so other purines and pyrimidines. What's unique about the cow is that orotic acid is virtually the only purine or pyrimidine found in the milk. There's a small amount of CMP found in the milk, a very small amount of AMP or cyclic AMP, and all the rest of it is orotic acid at fairly substantial levels. So the hypothesis for looking at milk orotic acid was that in non-ruminant species, orotic acid is known to be elevated in the urine when the urea cycle is blocked either by genetic defect or by deficiency of arginine. Willard Visick and John Milner were especially involved in this research. We hypothesized that perhaps the orotic acid in cow's milk might be a measure of nitrogen load in cattle. If we exceeded the capacity of the urea cycle to dispose of nitrogen, perhaps orotic acid in milk would be elevated. So our hypothesis to test for this hypothesis, we examined milk orotic acid in cows at graded levels of urea. At the time, Jimmy Clark was conducting an experiment assessing optimum levels of urea in cattle feed, and this urea would be converted to ammonia in the rumen, and we wanted to see whether, we piggybacked basically on this experiment to see whether these changes in urea level would lead to changes in orotic acid, milk orotic acid. We found that milk orotic acid did not respond to the levels of urea in the diet, but one of the cows tested, number 2778, had nearly 10 times more orotic acid than the others. This is cow 2778. She's the index case for DUMPS. We first studied her as a 6-year-old, and she lived to be nearly 14 years of age. She was not adversely affected by her condition, either in production, health, or reproduction. We examined the persistence of milk orotic acid in this cow, 2778, along with 8 cows that were normal for milk orotic acid. 8 cows that were normal had roughly 80 micrograms of orotic acid per milliliter, which was the value that had been in the literature. Our cow, 2778, had 5 to 10 times more orotic acid in her milk. In addition, during her lactation, she had elevated levels of orotic acid in urine and blood, although the levels were not nearly as high as they are in her milk. Uridine monophosphate synthase is the enzyme that metabolizes orotic acid and converts orotic acid to uridine monophosphate, UMP. This enzyme can be readily measured in red blood cells, and when we measured UMP synthase in the red blood cells of cow 2778, we found that she had half of the normal activity of UMP synthase. And when we measured it in some other cows with high milk orotic acid, the same observation was made. We're able to, while we measure the enzyme in red blood cells, we assume that the level of UMP synthase is equally decreased to about half in most tissues of the body, including the mammary gland where orotic acid is synthesized. Being able to measure UMP synthase in red blood cells permitted us to look at the level of this enzyme in non-lactating cows as well as in bulls. So this is the reaction that's catalyzed by uridine monophosphate synthase. Orotic acid is converted to uridine monophosphate or UMP. UMP subsequently is converted to the other pyrimidine nucleotides that are required for growth, development, and activities. A partial block in this enzyme would lead to a buildup in orotic acid, and this is what appears to be the case in this case.
So what happened with the DUMPS research? As you just heard, the high milk orotic acid was found by chance, and there were no treatment differences, although cow 2778 was high. Then Jim and Jimmy went on and sampled the other animals in the U of I herd, milk samples at this particular time to determine the milk orotic acid. They found 2 more cows with high levels of orotic acid. There was a graduate student in genetics that looked at it and said, well, there's no genetic component here. These, don't bother us with this. And it was about that time, shortly after that, that I got involved, and I took a look at the pedigrees, and I found that Skokie sensation Ned was in each of the 3 pedigrees of these animals. And that can be represented here on the pedigrees of the 3 families. Our X in the box is the common sire. This also points out that when we first did work in this area, we did not disclose the identity of the bull because we had not confirmed all the inheritance and consequences and what it was meaning. But here we have 2778. Her sire was in fact Skokie sensation Ned. Skokie sensation Ned was also the grandsire of 3365 and the great-grandsire of 3768. Now, part of my fascination with Skokie sensation Ned might have been that at that particular time I'd already milked some daughters of him, and he was bred at a farm that was about 20 miles or so from my house that I grew up at. Okay, more on these pedigrees. Then you can see that 2778 had 2 daughters, 4043 and 4459. Both were what turned out to be carriers, and a granddaughter, 4553, that was a carrier. 3365 had 2 daughters, neither that were carriers. They tested normal, and 3768 had a carrier offspring as well. Now, when this was published, some of these animals were immature. That's why the asterisks are on here. But as you can see from this particular picture of most of those cows were in a previous pedigree, they grew to be nice, mature cows. Hopefully the other thing you might be able to see from this picture, or what I should say, what you don't see from this picture, is if you just look at these cows, they're relatively normal. There's no indications that they may be DUMPS carriers or only being heterozygotes for this particular condition. And so that's one of the things that's where being able to measure the enzyme then after the milk orotate allowed us to differentiate this. So how did, as I mentioned, DUMPS they were measuring the enzyme, the UMP synthase. This was prior to DNA, so in terms of those tests being available, so that was not the option. And the other thing that we needed to do is we had what we perceived to be normal and heterozygous animals, but we really had to determine where's the break point between the normal and the heterozygous animals. And as already was mentioned, the activity of UMP synthase is about 3 on the normal animals and about 1.5 on the heterozygous animals. With the activity close to 3 for the normal animals, we'd also expect greater variability among those normal animals than we would have among the heterozygous animals. And so what I think an important concept that we applied here is we recognize the biological system. We equated the coefficient of variation between the normal animals and the heterozygous animals to help us determine what criteria we should use to define the animals in the 2 groups. And if you're ever doing enzyme activity testing in the future with a couple of groups, I encourage you as well to think about equating coefficient of variation rather than just looking at the average difference between 2 means. So our research proceeded, sampled the descendants of Ned, and again, Jim, with his concern about details, reminded me this morning that this sampling was first milk samples of different animals that were, and of the random herds in Illinois. Milk samples were sent into the lab, and the levels of milk orotic acid were determined. And then Jim and I had the opportunity to go visit lots of farms to get blood samples as well as milk samples and follow up on some of these animals. And these farms were across the Midwest. And this, again, we mentioned that this research was done prior to DNA. This research was also done prior to GPS. And so I was navigator and Jim was the designated driver, and we got everywhere safely. I can't say we got everywhere on time. I remember one particular case that we were supposed to get to a farmstead by 5 o'clock, and it was close to 9 o'clock by the time we pulled in the evening. When we finally pulled into the farmstead, the farmer had just gone in to have his late supper, but he came back out and helped us get the samples from the animals that we needed. But part of that, what that also says is that we had a lot of cooperation from the dairy producers in the Midwest on doing this project and had some interest in it. We greatly appreciated that. We also then, with the samples, they included milk orotic acid in some cases or blood in others. With the AI bulls, we were able to use some blood, but I think we also used semen primarily. No? Jim's telling me not. We just used blood. All right. See, I remember this well. That's why the two of us are here together. Then once we had the samples, we evaluated results from planned matings. We sampled the embryos from carrier-by-carrier matings to determine what the consequences were, and we evaluated other consequences. For instance, one of the consequences that we were concerned about is that if we're having loss of embryos, that's going to increase the calving interval for different animals. And so we were evaluating that, or if there was differences in milk production of the different groups. So what conclusions can we make about dumps? We'll define it as a lethal gene. The uppercase N allele for the normal allele, the lowercase n to define the deficient allele. We then can define 3 genotypes that we would be expecting to be happening. As I mentioned, the normal performance or normal activity of the homozygous for the normal was about 3 for UMP synthase. Heterozygous about half that, reduced activity about 1.5, but from appearances, they had normal performance. And the homozygous recessive, that was the lethal and died before birth, and as you already heard, in our research indicated that happened about 40 days after conception. What was the inheritance of dumps? So if you have a normal by normal mating, all the offspring are expected to be normal. That's what we found. If you have carrier by normal mating, about half are going to be normal, about half are going to be carriers. Again, that's close to what we found. If we have carrier by carrier matings, we're expecting a 1/4, 1/2, 1/4 distribution across the 3 genotypes. But what in fact appeared was a 1-to-2 ratio of normals to carriers. And this 1-to-2 ratio of normals to carriers is traditionally and historically a representation of having a lethal genotype. And the homozygous recessive, those quarter never made it to term, so that's why we didn't see them. So that's why we were left with the 1-to-2 ratio between the normal to heterozygous offspring. So we went around and sampled these cows. We found 15 carriers among 880 cows in these randomly selected Holstein herds in the state. So that was a 1.7% incidence, large enough for us to be concerned about the condition. When we sampled the bulls, the AI bulls, there were 4 of 287 Holstein bulls that were identified to be carriers, so 1.4%. And so again, there's some significance of concern about this condition in the breed. One of the other research aspects is we looked at pregnancies that were confirmed at 1 month. There were 31 of these. Of those 31 pregnancies, 9 of them led to calves that were homozygous for the normal allele. And so they had no evidence that they would be, they did not have the deficient allele for dumps at all. With the heterozygous calves, they had 1 more normal and 1 deficient allele, and there were 14 of those. And even though we had 31 pregnancies, when they were checked at approximately 1 month, there were 8 embryos that were lost with at calving. The other thing observed from this is that our observed numbers here are very close to our expectation in terms of the 1-to-2 to 1 ratio. And so that was reassuring as well, and confirmation that this is just a homozygous recessive condition. So more on dumps research, deficiency of uridine monophosphate synthase. We had sampled 15 leading Jersey bulls. They were all normal. I think I've heard recently that there has been an appearance of dumps in Jerseys. There were some cases where there was some Holstein blood that moved that direction. We sampled 102 leading Guernseys bulls. They were all normal, no indication of having dumps in Guernseys. We also measured UMP synthase, and it was detectable in both 15 and 35-day embryos. And it was in these embryos that we were, in fact, able to differentiate 3 different genotypes. So this is some confirmation that there was sufficient amount of the enzyme or other resources that I think Jim's going to elaborate in a few minutes about, of how this might have been to let these embryos survive even though they had this lethal condition. And I'm always interested, okay, how big was a 35-day embryo? I don't know if you can see this grid across the bottom here. It's in 5 centimeters. That's approximately 2 inches. So if you have the eyeball and the head here of this particular embryo, the back line coming out, and then you've got the tail and the rear legs together. So this embryo is approximately an inch from top to bottom, relatively small, but this was the size we were able to measure the activity in. Also, I should point out that, excuse me, this is a much smaller embryo than what would have been used for testing of mule foot, which had been done prior to our work with dumps.
So why would the full deficiency of UMP synthase be lethal? If we look here at the reaction catalyzed, orotic acid being converted to uridine monophosphate, a complete blockage of this reaction would mean that the embryo could not make uridine monophosphate and all the other pyrimidine nucleotides that are derived from it. These pyrimidine nucleotides are used for DNA, RNA, and other metabolic functions, and clearly without the ability to make uridine monophosphate, this condition would be lethal. But the more interesting question is, why do embryos survive for 40 days or so, even though they are totally deficient in this enzyme? And it seems to me that there are 2 possibilities to explain this. One is that in the fertilized ovum, there are a substantial amount of pyrimidines present, and that these are being recycled in the early part of embryonic development. And so the embryo can make use of the pyrimidines that are already there. Only when the demand for pyrimidines exceeds what would be present in the fertilized ovum do you have a situation where the embryo can no longer survive. The other possibility is that in the fertilized ovum, you do have some maternal, you do have maternal UMP synthase, which would be normal, or in the case of a deficient animal, half normal, which is sufficient for growth. And so it's possible that in that early phase that the maternal enzyme may be able to make some UMP synthase and hence the other pyrimidine nucleotides that are required for growth and development. There is an analogous condition in humans. In fact, it was known before we started our work. And it's called hereditary orotic aciduria. In the homozygous recessive condition, they also lead to embryonic mortality, although it's not been studied. But some homozygote recessives are in fact born and would die shortly after birth. That can be prevented if uridine is provided orally, promptly, and for the rest of their lives to these particular children. So in this case, if they're born, they're able and you're able to provide them with exogenous uridine, they do in fact survive. On the other hand, if this treatment is not started promptly, there are developmental problems, both physical and mental retardation, many of which are irreversible if the treatment is not started immediately. The question becomes, why do we not see any bovine homozygotes surviving to birth? And it seems to me that what we suspect is that the maternal provision of uridine to the fetus is able to occur in humans, but not in cattle. So that there's some placental transfer or placental generation of uridine to provide to the growing embryo in the case of the humans, but that is not seen in cattle. It would be something that would be interesting to study and to confirm.
So DUMPS was declared undesirable recessive by Holstein Association USA. This was an opportunity where Jim and I went and spoke to the Holstein board, identified what the inheritance was, identified the frequency in the population, identified the potential consequences, and recommended that they declare it an undesirable recessive and have an aggressive program to eliminate it from the breed. So AI organizations had their bulls tested, and heterozygotes were identified by DP for being a DUMPS carrier, and we'll also talk about TD a little bit later. I'll leave you in suspense until I get to that particular component. And the young bulls that were shown to be carriers, they were not permitted to undergo progeny testing. So obviously with this comment about progeny testing, this obviously was prior to the genome evaluations that are currently going on now. But at that particular time, progeny testing was the main way to determine the genetic evaluation of the bulls. So how extensive was the testing? Okay, so we've got the bars. The open bars are tested normal. Closed bars are the DUMPS carriers. You might notice that the scale is not equal dimensioned on the left side. But what hopefully you can get the picture from this is that when we started the official testing program of Holsteins, had just a few, my mouse disappeared. There we go. We had just a few carriers identified, then on a quarterly basis, then a few more, and 89, quite a few in the beginning of the year, continue to then get fewer and fewer carriers identified throughout the years subsequent to that. So these were identified and people were evaluating the pedigrees and not having them go further in their genetic evaluation. And if we summarize that same data but on an annual basis, I think you can see the same pictures, and there's one more year of data here as well. So where in '88 there were 65 heterozygotes or carriers identified, up to over 300 in the next year, down to just about 150 the next year, and then very small numbers the subsequent years after that in terms of carriers that were identified. And so we at this particular time, we decided that we did such a good job, we were putting ourselves out of business. But I think that was really for the benefit of the breed for that to happen. And that's also in contrast to some of the other policies that have occurred with other genetic disorders in the breed that have not been quite as aggressive, and they continue to be in the population and not be eliminated as DUMPS essentially was. All right, was it just 4 bulls that have this condition? Well, in '87, we'd identified the number 5 bull on the type performance NTP index as being a carrier, happy herd beautician. Also, mutual was identified in John Red, and I'll talk a little bit more about needle-lane John Red on the next slide. And happy herd beautician continued to be a high-ranking bull for the next half a dozen years. The other thing to notice is happy herd's beautician genetics did not change. But in '87, he was 5th rank, but then because of progress in the breed, in '88 he was down to 17th, in '89 down to 31st, and subsequently declining after that. So his decline in number here is not that his genetics changed, but his competition that he was being compared with greatly increased, and there was a lot of progress occurring in the breed. Okay, a little bit more then on needle-lane John Red. He's going to be identified as this Red Proband. And so the P in this box that's mostly black is identifying that he's a heterozygous male. And we were saying, okay, we didn't know that there had been any red DUMPS carriers prior to his detection. And so we were able to sample his sire. His sire was, in fact, known to be a DUMPS carrier. And then we had lots of offspring of his sire as well. For instance, there were 20 sons of Needle-Lane John Red sire. Those 20, so these are brothers to Needle-Lane John Red. 12 of them were carriers and 8 were normal. There were 11 sisters. 6 were carriers and 5 were normal. So this is very consistent with this sire being a carrier and randomly made it to the population. We go back on the female side of Needle-Lane John Red. We were not, the dam tested normal, and to confirm that, they had 2 male sons, and both of those were normal. Now we go back another generation to the grandparents. We weren't able to test any of the grandparents. However, we were able to test some progeny of the grandsire Needle-Lane John Red. 13 sons were normal, and 2 daughters were also normal. This is sufficient to say that with high probability, this grandsire is likely normal and not where the DUMPS condition came from. So we're looking over on the female side. Well, we actually had 2 offspring of her. Both of those were normal. But we need to recognize that those 2 normal offspring are not sufficient to rule out the possibility that this female is quite likely the carrier that transmitted it on to her son that then transmitted it on to Needle-Lane John Red. So this is just some of the, tried to walk you through of, we found a particular animal, then we were able to go back through the pedigree and it sorts through and follows the expected genetic inheritance. What was the timeline of DUMPS? Well, 1969 was a good year. 2778 was born. I started as a freshman at U of I, and Robinson joined the Dairy Science Department at the University of Illinois that same year as well. In 1983, the PNAS publication first was identifying the high milk orotic acid information. In 1984, DUMPS was named and the inheritance was published. And I say DUMPS was named, it's really easy to say, but Jim and I spent quite a bit of time going back and forth about possible names and what this should be called or not be called. And because we considered it not a very good condition, we wanted people to recognize that if they had animals that had this condition, they really should be in the DUMPS. And so that was just a secondary interpretation instead of the acronym that Jim had defined for you previously. Anyway, I should move on. He's laughing hysterically here to himself. In 87, it was declared an undesirable recessive, as I mentioned when we visited Holstein Association. In 1993, it was virtually eliminated from the breed. And so just because of the aggressive program, not having carriers be future sons, that was why it was able to be eliminated. We were not culling any of the females. I don't think I mentioned before, but if you had a female that was a carrier, she could be quite productive in her own right as long as you did not mate her to a carrier bull. And so just by controlling the male side, we're able to eliminate this condition and control its spread. Also in 93, the DNA mutation was identified and a DNA test was developed. And in 2015, we ended up giving a DUMPS webinar for the Graduate Student Division of ADSA. The current status of DUMPS and of many other inheritance issues for animals can be found in the OMIA, the Online Inheritance of Animals. I encourage you to look at that if you have a question about any genetic abnormality or unusual condition that you might run across in your research. But OMI 262 is the number for DUMPS, shows deficiency of uridine monophosphate synthase. Excuse me. The current status is some people decided, well, we need to have more consistent naming, and so you will also sometimes find DUMPS identified with the haplotype HHD. It's been found on chromosome 1, the region has been identified, And the current frequency of this condition is 0.01%. And so I really do think we did an excellent job of ridding the breed of this particular condition. And as I mentioned, one of the things that we had recommended to the Holstein board is when they labeled animals as carriers, as DP, they also needed to identify those animals that were tested free of the condition. And so we used the T for tested free and TD for tested free of dumps. And some of you might know that I'm a football fan, and so this obviously has positive connotations for me if that we're having a touchdown here, if the animals are tested free. And we have this information also to publish because it was just as important to identify the carriers, and it was important to identify those that have been tested and were known to be free of the condition within these pedigrees. And I think that was an important thing that we implemented as well. Another thing I'll mention on this is that our Holstein Association continues to use the T for other conditions that have been tested free of these conditions. So even though it started in dumps, it's now a practice of identification that's being used in many others.
Subsequent to our work, German colleagues identified the DNA defect that's present in dumps. The dumps mutation is a C to T substitution in the gene for UNP synthase. This mutation leads to a stop codon in the corresponding messenger RNA, and accordingly results in a truncated and apparently an enzyme, a protein that's devoid of activity. And this mutation has permitted the development of a DNA-based test for the condition, which simplifies things and permits semen to be evaluated as opposed to requiring a blood sample. After our work with dumps, we studied other inherited disorders. So the effect of dumps had a continuing effect. Citrullinemia is a defect of the urea cycle. It was discovered by Australian colleagues, and we were able to study its origin here among American Holstein bull, and we studied the prevalence of the condition in American Holsteins. For us, the problem in the U.S. Holstein population, the problem is essentially negligible. Unfortunately, in Australia, they used one of our bulls, and through a lot of inbreeding, developed a very high incidence of the condition. Another inherited disorder we studied was factor XI deficiency. This is a blood clotting disorder, and we helped to discover what the mutation was that led to this particular condition. The third inherited disorder that we studied was Escherichia coli. This is an intestinal tract blockage disorder, and we were able to establish the inheritance of the condition. So to review, serendipity and dumps, our herd happened to include Cal 2778. Cal 2778 happened to be included in those samples for a possible effect of urea on milk orotic acid. We had the good fortune to follow up on this oddity of high milk orotic acid, and we were able to discover this well-hidden undesirable recessive and to participate in this elimination from the Holstein breed, improving the Holstein breed by helping to eliminate this undesirable mutation. So what's the moral of the dump story? The first is, the main one is to be open to serendipity. Be aware that you may stumble upon something that's totally unexpected and may lead you into a very fruitful line of endeavor. But whether you're looking for a serendipity, one must be prepared and be able to recognize the serendipitous observation that one has and how one might be able to pursue it. As stated very well by Pasteur, in the field of observation, chance only favors the prepared mind. This is certainly the case for the dumps story. Before opening up for questions, we have some acknowledgments that we'd like to make. Financial and in-kind support were given to us for this work by the National Dairy Council, the USDA, both hatch and competitive grants. The Holstein Association of America was involved with this project from nearly the beginning. The various artificial insemination organizations were very keen to participate and to help us with this project. As Roger mentioned, the Midwest dairy farmers, dairy producers who permit us to sample their animals and to follow up on the condition is very much appreciated. The Division of Nutritional Sciences and the Department of Dairy Science, now the Department of Animal Science at the University of Illinois, were also very supportive of our research in this area. We'd like to also acknowledge some individuals. Among faculty, Bruce Larson first got us interested in milk orotic acid. Jimmy Clark allowed us to piggyback onto his urea feeding experiment. And Dale Nelson and the vet school was very important in terms of the flushing of embryos when it came time for that particular part of the experiment. We had a number of technicians involved with this work. As you can imagine, telling someone that their cow was a carrier for this condition, one had to be very, very careful that the experiments were done well and that we were absolutely certain that we were identifying a carrier animal, that in fact that animal was a carrier animal. A number of graduate students also cut their teeth on the DUMPS project, both master's and PhD students, and we very much appreciated their contributions. In addition, a number of undergraduate students participated in the experiments in terms of collections and doing some of the lab work. We very much appreciate the contributions by these individuals. We would now be very keen to open things up for questions, and if your particular question doesn't get asked today, please feel free to contact either Roger, Shanks, or me at the addresses indicated. Thank you very much.
All right, thank you both. That was a very interesting story, and I'm impressed at the, I guess, the financial consequences that disorder could have had for reproduction, from a reproduction standpoint. If you do have questions, type them in the Q&A box, and we'll get to them. We do have one from Ron Kinticker, the Ohio State University. He asked, do the male DUMPS carriers have any overt growth deficiencies?
Do the male DUMPS carriers have any what?
Any overt growth deficiencies?
No, not that I'm aware of. I don't think there were physical conditions there, but we did not directly measure the physical characteristics of the males, but I did not hear of anybody questioning any associations or anything corresponding to that.
And I think what that says is that half the normal level of uridine monophosphate is probably sufficient for growth, development, and sexual maturity, and all those kind of things. And that's probably the case. Most enzymes are present at more than the minimum level required, and that appears to be the case with DUMPS.
All right. I had one, I guess. I was interested to hear that you said the concentration of orotic acid in the milk was much higher than the plasma or the urine. Is there some, I guess, preferential uptake of orotic acid by the mammary, or is the mammary UMP synthase different somehow? Is that what I guess what's do you know the reason for that?
Yeah, we don't really know why normal cows have relatively high levels of orotic acid in their milk compared to any other species that's been examined. And so we don't know. I mean, it would seem like there must be some selective advantage to this that allowed this to develop. but we certainly have not been able to sort that out. It's conceivable that the orotic acid in cow's milk is some advantage to the calf being suckled, but once again, we've not been able to sort that out. And why is it that it's the mammary gland? I mean, the orotic acid is being synthesized in the mammary gland. That's quite clear from mammary cell culture experiments. So the mammary gland is generating this extra orotic acid, and then it's being secreted, seems preferentially and to the exclusion of other purines and pyrimidines, which are present in the milks of other species. So there's something unusual going on here, but we've not been able to sort it all out.
It's more than the cow misspelling the acid.
That's interesting. I guess I had one more question. Is there any consequence to this difference in orotic acid for humans when they consume cow milk, or is it just mainly a problem with reproduction?
It's time to start to.
Yeah.
Thank you for that question. A lot of the research that I did before we started working on dumps was, in fact, related exactly to the question that you asked. Because people will say, well, you know, since there's a lot of this orotic acid in cow's milk and there isn't in human milk, is this a deleterious condition? And it turns out that the response to orotic acid is highly species specific. So if you feed orotic acid to rats, they develop a fatty liver within a very short period of time. But it turns out that's unique to the rat. All the other species we've tested, from other rodents to guinea pigs, hamsters, monkeys, the other species we tested, none of them responded in that fashion. And to the extent that we looked at it, but not looking at fatty liver, but looking at fat at lipids in the blood, it doesn't seem to be a problem in humans either. But it is in most infant formulas, they do add these other purines and pyrimidines to them at these low levels, but it's not clear that makes a significant impact. So I hope that helps you answer the question.
Yeah, definitely. Okay, Gabriella Maldini has a question. So what is the standard test to identify carrier bulls, and how much does it cost?
Okay. The standard, I don't have all that right in front of me, but now most of the bulls are going to have their genome evaluated, and the DUMPS test can be an additional consequence of having their genome evaluated. On a female side, though, my recollection is that the cost for the DUMPS test was approximately $40, although I'm thinking it might have gone down from that. But that's what it had been the last time I looked at it. If you're wanting to have information about what of testing, I would encourage you to go to the Holstein USA website, and they have the details about what the cost is for DUMPS, as well as some other genetic abnormalities in cattle, being BLAD or CVM or some of those others. One of the things that Jim and I tried to do when we're setting up DUMPS is make it publicly available, those tests in the public domain, rather than trying to set up any patents or do something along those lines. And so those tests should be readily available to be included. At this point, the frequency is relatively low, and so if I was going to be, in general saying, okay, should we go out and test for DUMPS, I would say more to think about, okay, first take a look at the pedigree. and decide, you know, is this something that's recent? Is there a carrier that's known to be close up in the pedigree? And then you might consider testing for it. Although I will admit that I know that the recent cases of dumps that they have identified have been 4, 5, 6 generations from a previously known carrier. So it can remain in the line, if you would, for a number of generations without it being detected. Was there another aspect of that question, Eric?
No, she said thank you.
Okay.
So you did say that this was a C to T mutation, so this could just occur spontaneously in a cow then, right?
Well, we have no evidence that it recently occurred spontaneously. And by that I'm meaning that our known cases of dumps we can go back in the pedigree and in many cases find Skokie sensation Ned or some of his ancestors that may or may not have been carriers in those pedigrees. And so we're not aware of an additional mutation of the same thing happening besides that family line.
I see. Well, I don't...
Jim's got something else to add here.
Well, just want to mention, in terms of the human condition is characterized by a variety of different genetic mutations that lead to the condition. But in the case of cattle and other livestock species where the animal is being bred for production, you're likely to find that it's one particular mutation that is passed on along with the desirable features or genetics of a particular animal. But it's certainly possible that you could have a new mutation in dumps, but there's no evidence of that at this point.
Okay. Well, I am not seeing any other questions, so I'd like to thank you both again for taking the time to tell this interesting story, and remind everybody that if you have any questions, any additional questions, feel free to get in touch with Dr. Shanks and Dr. Robinson and via their email addresses there. So thank you all for your attendance, and thank you again, Dr. Shanks and Robinson. And
thank you very much for inviting us. We greatly enjoyed giving the presentation.
Thanks again.
Yep. So I guess that will conclude the webinar. Look forward to the next one. Keep your eye out for emails, and thank you everyone for attending.